First a few words by Tiffany House. Yes, Tiffany?
Hi, everyone. I want to thank you again for being here. I think we had a great day yesterday. We learned kind of where we've been, a lot of the history of ERT. The roundtable was fantastic. I think, we learned a lot from each other. We learned a lot about the different approaches being taken in different countries. And now is the really exciting part. Now we're talking about what's the future research for our disease, what's coming next.
Thank you, Tiffany. So you've seen today's program. It's I called it ongoing research to build the future, because future research, you never know what kind of research you need tomorrow. The first speaker is Dr. Nina Raben, and her topic will be pathology. Huh? Oh, put up a scene.
First, I also would like to thank the organizer, Masha Zimmerman, and in particular Tiffany, for inviting us and for organizing this wonderful meeting. It is now well established that accumulation of undegraded substrate in the lysosome, in Pompe disease, as well as in other lysosomal storage diseases, leads to multiple secondary abnormalities. And the complexity of the pathogenic cascade in lysosomal storage disorders indicates that successful therapy will require a combination of drugs to address different aspects of the pathogenesis rather than a magic bullet designed to address the primary defect. So we're looking at these different aspects of pathogenesis to try to identify new points of therapeutic intervention. We have found that in addition to enlarged glycogen-filled lysosome, there are multiple secondary abnormalities in Pompe skeletal muscle. We found defective autophagy, a major secondary abnormalities in Pompe disease. Defective autophagy is a very common feature in many lysosomal storage disorders, which actually gave rise to the idea to call these diseases autophagy disorders. We've also found disturbance of calcium homeostasis in Pompe skeletal muscle, multiple mitochondrial abnormalities with reduced ATP production. But today, these data are published. Today, I will talk about new ways to overcome lysosomal and autophagic defects, and about dysregulation of mTOR signaling pathway in Pompe skeletal muscle. As I've told before, for these studies to look at the different aspects of the pathogenesis, we use our mouse model of the disease. And we also use the recently developed and very much needed cellular model of the disease. These are immortalized myoblasts derived from Pompe mice. Oops. And these myoblasts, when differentiated into multinuclear myotubes, are filled with enlarged lysosomes. As shown here, the lysosomes are in red. We have found that in Pompe skeletal muscle, in addition to enlarged lysosomes, as is typical for Pompe disease, we see large areas of autophagic accumulation. Very briefly, autophagy involves formation of double membrane, which engulfs a portion of the cytoplasm, leading to formation of a double membrane vesicle autophagosome. Autophagosomes fuse with lysosomes where the content of autophagosome is digested and recycled. And in fact, we see classical double membrane structures by electron microscopy, these autophagosomes filled with undigested cytosolic content, and those autophagosomes pile up in the vicinity of lysosome, suggesting that the fusion between these vesicles is impaired. And in fact, when we did the time-lapse confocal microscopy, oops, of the live muscle fibers in which both autophagosomes and lysosomes are labeled, lysosomes are here in red and autophagosomes are in green, we were able to directly show that indeed the fusion between these vesicles is impaired, and this leads to accumulation of autophagic debris. To restore this vesicular fusion, we took advantage of the data by Andrea Balabio in Italy, who showed that the transcription factor EB, TFAB, is a master regulator of lysosomal and autophagosomal biogenesis. This transcription factor, when moved, translocates to the nucleus and is activated, it binds to clear sites in the promoter region of the autophagic and lysosomal genes. CLEAR stands for coordinated lysosomal expression and regulation, and stimulates the biogenesis of both autophagosomes and lysosomes. This is quite a remarkable finding because it elevates the role of lysosomes. So the lysosomes are not just the dumping site of the autophagosomal content, but they are an active component of the autophagic process. So both autophagosomes and lysosomes are coordinately upregulated. And it makes a lot of sense because if you have an increase in the number of autophagosomes, it may overwhelm the system. And this mismatch between the number of autophagosomes and lysosomes may have a dire consequences for the cells. What they also showed that in addition to stimulation of autophagosomal lysosomal biogenesis, activation of this transcription factor stimulates autophagosomal lysosomal fusion and stimulates lysosomal exocytosis. Lysosomal exocytosis is a process by which lysosome fuse with plasma membrane and discharge the content of lysosomes outside the cells. Stimulation of lysosomal exocytosis is a new, conceptually new therapeutic avenue for lysosomal storage disorders. And when we expressed this transcription factor in Pompe skeletal muscle, the effect was quite dramatic. On the left, you see the Pompe skeletal muscle fibers filled with lysosomes and autophagosomes, and on the right, you see the Pompe fiber transfected with TFA3, and it resulted in a dramatic reduction in the number of large lysosomes, and it alleviates the autophagic buildup. Our data in collaboration with Rosa Portolana at NLHBI showed that another transcription factor, which is a member of the same family of transcription factor, is another master regulator of lysosomal and autophagic biogenesis. And I used to joke that we're now that we're slaves of two masters now. This transcription factor is TFE3. And it seems that this is a better target in Pompe disease because it is abundant in skeletal muscle, whereas TFAB is barely detectable, as was shown here by Western analysis and by RNA sequencing. We have shown that the downstream targets of this transcription factor are very similar to those for TFAB. And as you can see here, this is a cheap-seq analysis, which shows that both lysosomal and autophagic genes are downstream targets for these transcription factors. And this was done in muscle cells, C2, C12 cells, and our immortalized wild-type myoblast. In addition, and quite unexpectedly, we also found that the large group of target genes constituted mitochondrial genes. And this feature of TFA3 in stimulation of mitochondrial biogenesis may actually be very helpful in Pompe disease because, as I mentioned before, there are multiple mitochondrial abnormalities in Pompe skeletal muscle. Overexpression of TFA3 in Pompe muscle cells reduced lysosomal size, reduced glycogen accumulation, and it did it through stimulation of lysosomal exocytosis. As you can see here, LAMP, the marker for lysosomes, which is normally seen inside the cells, can now be seen outside the cells. Overexpression of transcription factors is a good proof of principle, and the overexpression may come at a price. Some muscle fibers show membrane detachment, and as you can see here, the lysosome, which is shown in red, is literally moving toward the plasma membrane. And occasional fibers with high level of overexpression of transcription factor shows really a striking membrane detachment, perhaps because of the massive lysosomal exocytosis. So for therapeutic purposes, one would want to activate the endogenous transcription factor rather than overexpress it. And again, this is a collaboration with Rosa Portolana. We found two compounds, which we for now call compound X and compound Y. And we've shown that these compounds activate endogenous TFA3 transcription factor. At low dosages, you can see the realignment of lysosomes along the plasma membrane, which is a telltale sign of the effect of the transcription factor. And it is actually very similar to what you can see when the TFAB, for example, is overexpressed in skeletal muscle. In higher dosages, compound X stimulates translocation of the transcription factor into the nuclei where the transcription factor is activated. Compound Y behaves the same way, perhaps it is less robust. And what is important here, that these cells are cleared of enlarged lysosomes, indicating that they are phenotypically corrected. I will now move to the second part of my talk about the dysregulation of mTOR signaling. Yes, one thing which I forgot to say, that we're planning to test these compounds in vivo in the knockout mice. So why study mTOR signaling pathway in Pompe skeletal muscle? mTOR is a mechanistic target of rapamycin complex 1, is a master regulator of protein synthesis, and it is directly involved in the control of muscle mass. Pompe disease is a muscle wasting disorder. Muscle atrophy is a hallmark of Pompe disease, both in humans and in the knockout. You can appreciate the difference in size of the fibers in 18-month-old knockout mice. So even if we are successful in clearing the cells of enlarged lysosomes and autophagic debris, muscle atrophy will still remain and will still pose a significant problem. Another reason to study mTOR signaling pathway is because the recent advances in the lysosomal biology show that this activation of this transcription factor happens at the lysosome, that the lysosome is a platform where this mTOR kinase is activated. And I have to say, in particular for non-scientists, that this whole part of my talk is pretty complicated. So please, it's kind of hard to follow. Please don't be discouraged, because it took me almost 2 years to figure it out. So what was shown was that at the lysosome, RAG complex is in active configuration. Under nutrient-rich condition, this RAG GTPases recruit mTOR from the cytoplasm to the lysosomes, where it is activated by another protein, Rab, which resides on the lysosomal membrane. Once activated, mTOR phosphorylates its downstream targets, stimulates protein synthesis, and inhibits autophagy. We have found that the phosphorylation of 2 downstream targets of mTOR is significantly decreased in Pompe skeletal muscle, and we showed it both in whole muscle and in myotubes. This is consistent with inhibition of protein synthesis in Pompe skeletal muscle. The inhibition of protein synthesis was confirmed by another assay with puromycin assay, and as shown here in the knockout myotubes, the protein synthesis is decreased, as you can see, by a less intense bands in the knockout by Western analysis. However, it looked that the recruitment of mTOR to the lysosome was not affected, that this process goes fine. Because when we immunostained myotubes with lysosomal marker LAMP, which is in red, and they stained it for mTOR, these two co-localized both in the wild type and in the knockout. Again, indicating that the recruitment of mTOR to the lysosomes is not affected. So the next obvious candidate to look at was this Rab protein. And we found a reduced activity of Rab in Pompe skeletal muscle, as we showed by Western analysis. We then looked at the multiple proteins upstream from mTOR, which are involved in protein synthesis and degradation and autophagy. And we found that 2 powerful inhibitors of mTOR, PRS40 and TSC2, are increased and activated. The upstream kinase, AKT, which phosphorylates and relieves the inhibitory effect of PRS40 on mTOR, did not change. Therefore, we focused on TSC2 because its activation fits with the activation of its upstream kinase AMPK and LKB, and because TSC2 inhibits mTOR by inhibiting Rab. And furthermore, the whole cascade of AMPK activation of TSC2 followed by inhibition of Rab happens again at the lysosomal membrane under the condition of low energy, which seems to be the case in Pompe skeletal muscle. Indeed, we see the increase of TSC2, LKB, and phosphorylated AMPK on the lysosomal fraction, and we see a significant increase in ADP-ATP ratio in Pompe skeletal muscle, again indicating the low energy condition. However, under this low energy condition, normally mTOR should be away from lysosomes, and instead, as I showed previously, at this condition, mTOR still remains on the lysosomes, as shown here by immunostaining and again by Western analysis. If anything, that there is increase in mTOR in the lysosomal fraction. We then looked at the condition of amino acid starvation. And in the wild-type myotubes, as expected, after 2 hours of amino acid starvation, mTOR moves away from lysosome. You can see much more pure red in the wild type. And in contrast, in the knockout, even after condition of amino acid starvation, mTOR still remains on the lysosomes. And under this condition, we can see a further increase in AMPK and TSC. So this is a very unusual localization of the mTOR in Pompe mice. And the question in Pompe muscle cells. And the question we asked whether it is related to the lysosomal size. And then we did the experiment in myoblasts, which do not replicate lysosomal pathology. Myoblasts do not have enlarged lysosomes. And we found that in the myoblast, after amino acid starvation, as expected, mTOR moves away from the lysosomes, and again, you see more of a red color here. So, which suggests that this unusual and very disturbed localization and activity of mTOR is linked to the enlargement of lysosomes. So here is our model. So we think that the enlargement of the lysosomes, and the lysosomes are filled with glycogen, and our previous experiment showed that they have increased pH. This lysosome no longer maintain their normal acidic pH. This results in the activation of vacuolar ATPase, which pump the protons into the lysosome. Essentially, the vacuolar ATPase is in overdrive. When this vacuolar ATPase is in overdrive, it results in an activation of Rab GTPases. This in turn results in the recruitment of mTOR to the lysosome, but on the lysosomes, it's not fully active because this part of the equation is also affected because of the inactivation of TSC followed by inhibition of Rab. Under amino acid starvation, the left part stays the same. The mTOR still remains on the lysosome, and despite even higher levels of TSC and AMPK on the lysosome, mTOR is still not fully inactivated. So what can be done about it? There are two possible approaches. One is to inhibit TSC2 so that the mTOR will be activated. And this is our goal, to activate mTOR. And the second, and perhaps even smarter approach, is to try to acidify the lysosome. If we can decrease lysosomal pH, we will allow mTOR to move freely to and away from the lysosome so that it can be properly regulated. And we are currently exploring the possibility of acidifying this lysosome with acidic nanoparticles which are available. Thank you very much. And I would like to thank people in my very small Pompe group, 2 postdocs, certainly Rosa Poretolana, with whom we had a wonderful collaboration, Andrea Balabio and Giancarlo Parenti, and also Genzyme Corporation and Acid Maltase Deficiency for their very generous support. Thank you.
Thank you, Dr. Raben. A very small group indeed, but very powerful research and very, very impressive. And we do need to have this information to understand pathology and find ways to a cure. The share of this session is going to be taken over by Dr. Raben, so she will announce and guide the other speakers.
So it is, okay, it is my pleasure to present the next speaker, whose last name I will never be able to pronounce correctly.
Pijnappel.
Thank you so much.
So, first of all, thank the organizers for this opportunity to present this work. It's a very exciting meeting, and I will talk here about muscle stem cells, where we did some work on Pompe disease. And I will start with some of the basics to explain what this is all about. And I would like to start with this little movie, which is
away cleanly the first time, a great start.
Right. This was the Olympics, and you see here the. So it's a very sensitive mouse, I see. So you go back. Okay. So my question to you is actually, what could be the effect of running on the muscles? I think this is a basic question, but can anyone help me? What do you think is happening to the muscles if these guys are running this fast? Sorry? Yeah, it causes damage to the muscles. So, and still these guys recover, and then they can do it again. So, just to trigger your mind, I will talk about the problem in Pompe disease and muscle repair. And as you might guess, stem cells are important here. And I will try to hint what we are aiming at to go to the clinic, maybe in some of the future. First, here you see a cartoon of a muscle, and you see, let's try this again. Yeah, you see here that the muscle actually contains muscle fibers. And if you make a cross-section here, you can see here fibers which have multinuclei and are arranged like this. And these are longitudinal structures. And these are the, many of you have probably got a muscle biopsy. And this is done to evaluate the pathology and to confirm the diagnosis. But also it is very useful for research, as I will show later here. And just to show an example how that looks. So on the left side you see a healthy muscle biopsy. So here you see what I just showed in the cartoon. These are the muscle fibers and beautiful arranged red circular structures. And here you see in blue, these are the nuclei. So there's the DNA. And the muscle cell, so one fiber is one cell, is special. It has multiple nuclei. You don't see that very often in other cells, but you do see it in muscle cells. So these are two examples from diseased, Pompe diseased patients. Here a classic infantile and here an adult onset. And you can see what's going on here. This is a strongly destroyed structure, and it's very severe. And in the adult onset here you see actually vacuoles, so muscle fibers are disappearing and vacuoles are appearing. And as you all know, this is caused by the accumulation of sugar chains or glycogen in these fibers. So, yeah, I think you already got the answer. Can damaged muscle recover? Yes, it can. this is a quite dramatic example that this is actually possible. So we have injured here a muscle in a mouse, and it can be a wild-type mouse here, and using a snake toxin actually, or barium chloride. You can use several agents to do this. And you can see here the response to that in time. So this is an enormous mess, of course. You can see that a lot of the fibers are destroyed, and there are a lot of nuclei. This is infiltration with immune cells. And this looks like a war zone, and you cannot imagine that this would be completely repaired. However, if you go in time, 5 days, 7 days, 15 days, here you see the final result. Again, a beautiful structure, beautiful muscle fibers here. And actually they got a central nucleus here that actually tells you that the muscle has regenerated. So new fibers can be completely formed in this injured muscle. So this shows the power of skeletal muscle and its regeneration capacity, which is very important. Of course, the question is, how is this possible? And this is possible by stem cells. So there are stem cells waiting in skeletal muscle. Normally they are just waiting and doing nothing. And when there's injury, they can act, they become activated, and then they start to repair. And here again a cartoon. I just showed you this. This is one cell, a cross-section of a muscle cell, and here you see the nuclei. And then there's a membrane around the cell, but there's another membrane here called the basal lamina, and the satellite cell, as it is called, or the muscle stem cell, is in between those two layers. And it's waiting there. As soon as there's injury, the satellite cell can move into the fiber and then start to repair. But it can also form a completely new fiber. We have actually stained, it's not so easy, they are quite rare, but you can stain these cells in muscle biopsies, and we have done that in controls and Pompe patients. So here in yellow you see two examples. So this is a muscle fiber. Here you see in green the basal lamina, in blue you see nuclei, and then here in pink you see the satellite cells. So there's a marker protein called PAC7, and you can use that to visualize satellite cells, muscle stem cells. And of course we were very interested in this basic question: what happens with satellite cells or muscle stem cells in Pompe disease? And this is actually relevant for all muscle diseases. Why can't they repair properly skeletal muscle? So we have used quite some biopsies from controls, as far as we had them, and Pompe patients, and we also looked at patients with Duchenne muscular dystrophy or DMD, just for comparison. And we looked in classic infantile patients, childhood onset, mildly affected adult onset, and severely affected adult onset patients. And we quantified the number of satellite cells in biopsies using the staining I just showed. And you can then express that per surface area. And it is known that when you are very young as a baby, you have more satellite cells. And when you get older, the amount of satellite cells is reduced. In green, you see the controls. And in red, you see the different Pompe groups. And what you can actually see is that there's no real difference between Pompe and controls. And also here, the Duchenne are here. This is an important message because there's a whole bunch of literature on Duchenne muscular dystrophy, and one of the ideas was that maybe satellite cells become exhausted or depleted during the disease due to continuous activation and continuous repair. So the question here was, are they exhausted? Are they still there? What is happening with them? Are they active? Are they inactive? And the first conclusion here is that they are there are satellite cells, there are muscle stem cells, and they are not depleted. So the next question was, are they active or not? And you can, actually a stem cell can do two things. Of course, if it becomes activated, it will divide. And then you get different outcomes. It can divide to make more satellite cells, so it can proliferate. or it can divide and proliferate and differentiate. So it can make a differentiated cell that goes into a muscle. So the muscle stem cell is undifferentiated and is a stem cell. So to look at proliferation here, we used another marker called Ki67, and that marks proliferating satellite cells or proliferating cells. If you do a double stain with PAC7, you can see if that cell was proliferating or not. And here you see an example of this cell is positive for PAC7 and it's positive for Ki67. So this one is proliferating. And here there's another one, slightly bigger, which is positive for PAC7, but it's negative for Ki67. So this is how we did it. The numbers are quite low. And what you can see here is a quantification of that. So this is actually the fraction of satellite cells that is proliferating as a percentage. And here you see the different groups, and again in green the controls, in red the Pompes, and here there's one Duchenne patient. And there's quite some variation between the different individuals, and this is actually known. Actually also for the numbers, it is very important to realize that you need some numbers to draw conclusions here, and that the variation between individual people is can be quite big. So that's really important. But there's no obvious difference here in the activation state between Pompes and controls. So that actually means that there's no extra activation of muscle stem cells in Pompe patients. So then we went on and we looked at muscle regeneration. You can actually detect muscle regeneration using a marker called embryonic myosin heavy chain, which you do see here in red. And maybe I should first go to this picture. This is a Duchenne. These are two Duchenne patients. You see the muscle fibers, and in red you see fibers that express embryonic myosin heavy chain. So this is a marker that is expressed in regenerating fibers. You can see clearly here high expression of this marker in several fibers, indicating that these are actively regenerating. But if you now go back to the Pompe biopsies, you can already see that here in the childhood or the adult onset patients, there's nothing that can be detected. It's not regenerating. In the classic infantile patients, these are two different patients. There's one here that shows a weak expression of this marker, and there's another patient which shows no expression whatsoever. So this actually shows that in Pompe disease, muscle stem cells are present, but they are not working properly. They are not doing their job, and they are not regenerating. And that's what you see here. And that is reflected in the level of muscle regeneration, which is quite low. And this also implicates that the machinery itself is there, and in principle could work if you might be able to activate it using another signal. So what we think is that somehow it should be possible to use this and to activate endogenous satellite cells or muscle stem cells to increase regeneration and to force the muscle to regenerate and to make new muscle. Right. So I switch now to another part. So to further investigate this, how can you actually investigate the activation of muscle stem cells, satellite cells? You need model system, as Nina just showed you, and one of the problems that we have with muscle cells is that they don't like to be cultured. Human muscle cells, they don't like to be cultured in the laboratory. So they stop growing quite fast, quite soon. As soon as you have a muscle biopsy and you take the muscle cells, you try to culture them, it's very difficult, and also the differentiation. So therefore, either you can immortalize them, as Nina showed, or you can do something else, which I talk about here. This is actually what I just said. So it's very difficult from a muscle biopsy to grow muscle cells and to do further research. And happily, there's another source, and it's called iPS cells. And this is Mr. Yamanaka, and he did a major discovery for which he got a very fast Nobel Prize in 2012 after the discovery in 2006 that you can make cells from other cells, you can make stem cells from normal cells. Nobody would have expected that this is possible. Normally you have a stem cell and you get a differentiated cell. He went actually back in time and made a stem cell from a differentiated cell using a very simple procedure actually. What is the implication? The implication is that you can take a patient cell, a fibroblast for instance, and you can, in the laboratory, convert this to an iPS cell, which is a cell that can be anything of the human body. So you can use this to make muscle cells, but if you have another disease, you can also use it to make other types of cells and study the disease in the laboratory. So this is an enormous advantage. And yeah, you can call this a patient in a dish. Actually, you have that patient, you have the genetic material, the exact genetic environment of that patient. You can actually model it in the laboratory, and that's an enormous advantage. So you can also test drugs for that. And what do you need? You just need some cells, so you can take fibroblasts or blood cells or whatever. So we actually did some work on this, and this shows you human iPS cells, in which we also looked at the mechanism of this process, which is on its own very interesting scientifically, of course. How does this work? How can you reprogram cells, which I will not go in here. But our major question to model Pompe disease was, can we make skeletal muscle cells? Can we make stem cells, muscle stem cells from it? And that took us a lot of blood and tears and sweat, how you call it in Dutch. But finally it worked with the help of some nice papers also published by others. And finally we were able, actually using a 35-day protocol. You have to imagine in human embryos or human adults, I mean, it takes a while to get these cells. We have to do it in the laboratory, and we're trying to mimic this. So this is quite a task. But in 35 days, you can actually make muscle stem cells in the laboratory from iPS cells, from skin cells, from patients. And that's shown here. And these are the red cells in this mixture. You always get a mixture, you have to purify, so this is what we did actually. And then you can differentiate them further, and then you get your long tubes, what you see here, that can be stained for markers for differentiated muscle cells. So we were extremely happy that this now finally worked, and now we can model Pompe disease in a dish. So this is important now because we can study the disease using this model system. We can use it for drug testing, for testing new therapies which we are working on. And another potential future application would be regenerative medicine, where you would use stem cells to replace muscle that is really gone in patients. This is a long way, of course, but at some point this may be an interesting application. So in summary here, we have seen that damaged muscle can strongly regenerate, has a strong capacity to regenerate in principle, and that is mediated by muscle stem cells. In a mouse, if you take away the satellite cells, this process is completely blocked, so these are essential cells. In Pompe disease, they are present, but they fail to do the job. We can make them from a skin biopsy in the laboratory via iPS cells. And these are now used in the lab for testing therapies. And we are trying also to transplant them and to see if we can replace muscle that is already gone. And I wanted to end here with all the people here in the lab that are involved in several projects. I'm happy to take questions. Thank you. Oh no, there are no questions, right? No questions. Sorry.
Okay, and the next talk will be on the GA genotype by Arnold Reuser.
Okay, all set. This is about genotype and phenotype. So what does the genetic information tell us about the disease of the patient, the phenotype? I'm a biochemist, and I came from the Department of Clinical Genetics. I'm now retired, but I still have a key of the lab. So I can enter the garage and everything, it's fine. Here you see two buildings. Here, this one was the old setting. This is the faculty tower where I worked for 40 years. And this is the hospital. And this was 1973 when I started. And now it's almost finished. It looks like this. So it was completely rebuilt. How do you do such a thing? You need a blueprint. And the blueprint tells you how to make the building. Right? So this is a blueprint there. And then you change the blueprint, you get another building. So the blueprint in the human is the DNA, the genetics. And the product, the building, is the GAA. That's the acid alpha glucosidase. So the DNA as a blueprint is the informative archive that you don't want to touch. You want to keep it intact. Then there is something in between. It's called the RNA. It's the information carrier. You know, if the DNA is the book is so precious that you don't want to give it out to anyone. So when someone asks for the book, you make copies. You make 10 copies, and from those copies, they are called RNA, and then you make the protein. So the book remains closed, the book, the DNA, the genetics, 25,000 genes, I showed you yesterday. Then we copy it. The monks did that by hand. It's called transcribere, you transcribe. So you have a copy, but the copy has exactly the same information. And the information is by the letter A, T, G, C. So you have the copy, and based on this code, letter code, you make the protein, and here you have the 40,000 products, and only one of these products is acid alpha glucosidase. Now, I tried to explain that these letters need to be, because it's a code, in exactly the right order. Now, for instance, I don't know whether I'm correct with translating the code, but it doesn't matter. You see a T, a G, and a C. And the three together tell you that at this position, there should be leucine. Now, what happens if it is not T, G, C, but T, G, G? Well, in this case, you still get leucine, because there are 6 different codes to make leucine. But for instance, if you would replace the C by an A, you have TGA. And that's a code that says, stop reading, don't make any more protein. So there's a big difference what type of change. And then, for instance, you have the ATG. What happens if you take out a T? Then first the triplets was ATG, and now it's AGC. So from there on, everything is different. If you insert one letter, the same problem. If you take out 2 letters, the same problem. If you take out 3 letters, you're missing only 1 amino acid, but the rest is still fine, because you did not change the code. All right, though. How risky is it really? How safe is it? Genotype-phenotype correlation. The DNA, it's very well protected, but nevertheless, it's vulnerable to lesion. I didn't know this, but when I prepared this lecture, I found out an estimated 1 million lesions occur every day. Every day. Changes in the genetic code, but virtually all are fixed by DNA repair systems. Unbelievable. All right, so we have this. Another Nobel Prize. We just had the 2012. Now we have this year's Nobel Prize. It went to in chemistry, and it was awarded to 3 scientists, and they discovered what systems made all these repairs in the DNA. So there they are, Thomas Lindell, Paul Modric, and Ashish Sankar. And they found out how the DNA lesions are fixed continuously. But if a lesion is not fixed, then it's called a mutation, and the mutation persists. And if you change the gene, you cause changes in the product. It can be a change to the better, but it can also be a change to the worse. Start with the better. Mutations to the better have led to evolution of, that's a difficult word, the Lopitaceus afarensis, has led to the 21st century humans, and that has taken 3.2 million years. Changes by changes by changes, without significant loss of function. All right, we go back. All right, so the DNA, the gene. So all these mutations cause different changes. If you have no mutation, what is it? No mutation. You have 100% production of acid alpha glucosidase. So that's fine. And the product is okay too, so you have 100% activity. You're not a patient. If you have a mutation that can lead to loss of production, so you don't make any alpha glucosidase, and you lose all your activity, of course. Another mutation, you make too little. Another mutation, you make the normal amount, but the GAA doesn't work, doesn't degrade the glycogen to glucose. Mutation number 4, you have little production and little function. And all these mutations, they are there in the acid alpha glucosidase gene, and altogether 500. They all have different effects. But at the end, they all lead to shortage. They all lead to shortage of GAA activity, and if you do not have enough GAA activity, you start to store glycogen, and you develop Pompe disease. Then if you want to make a genotype-phenotype correlation, we don't only need the genes, but of course you also need the patients. Then there's the hypothesis. If you have more shortage, you have a more severe phenotype. And that seems kind of logic. But it is a hypothesis, unproven. So this is the clinical spectrum. Patients with Pompe disease, they are born with Pompe disease, but the disease does not express immediately in all cases. In the very severe cases, which is here called classic infantile, the phenotype comes out very, very shortly after birth and is very, very severe with short survival. Sometimes onset of symptoms during childhood and sometimes during adulthood. So the hypothesis again, more shortage leads to more severe phenotype, but this is the hypothesis, but is this true? Now we go back to the skin fibroblast, the surgical removal of skin. Well, I'm pretty old. I can tell you, at this age I had, I think, up to 100 skin biopsies myself. It started when I was very young. You know, I tried to walk and I stumbled and I hit my knee on the street, it started to bleed, and a little piece of skin came off. Later, I started to use a knife, a scissors. I was drilling, there was an electric drill, and some people even use a chainsaw, you know. And it's all far more invasive than a skin biopsy because you do not notice it at all. Okay. So in my place, we started to collect skin biopsies in 1966. And when I started to work in 1973, already there was a nice collection of skin biopsies. And therefore we were able to make this slide. And this slide shows if you have skin biopsies, skin cells, this is what you normally find for GAA activity. There's a wide range. It is from about 40 to 160. So not all of us have the same activity. We're all different. So even healthy people don't have the same activity. If you then look at patients with classic infantile Pompe disease, very severely affected, they have very little to no activity. In childhood Pompe disease, you may be able to find some activity, but in some patients here, you don't find any. But still, they have a less severe phenotype. And adults, the large majority, they do have a substantial amount of residual activity, 5 to 25%. Here we have set, some have very little. Here we have set the limit of 30% average normal. And if we do a diagnosis using fibroblasts, because in blood spots doesn't work, in leukocytes it's difficult, but in fibroblasts it's perfect. If you find more than 30%, we say this is not a patient. It may be a carrier, so a parent of a patient, but it's not a patient. Okay, then here we have started to collect all this information about the 500 different mutations. It's all on the web. So you go to the www.pompecenter.nl, and here I have made a selection. It goes by the number of the letter, because in total the acid alpha-glucosidase genetic information, it's 3,640 something letters to link 956 amino acids in proper order. And here you have a listing. And then, so it goes by the number. You go from 3, 18, 54, letter number 118, and so on and on and on. And here we are at the end, 2,815. These are all abnormalities, changes in the DNA that we found. And then simply by looking at the genetic code, you can predict what it does on the amino acids. And here, for instance, you can see if you here remove 18 to 25, so you're removing 7 letters. And the code is by 3 letters. So if you take out 7, you get a shift. And that's what we call a frame FS, a frame shift. And then protein production stops. So that's very severe, and it's a P because it's predictable. You don't have to investigate, you know for sure. Here, for instance, I mentioned that to position 54, a change from a letter C to a letter T, but as I told you, you can code for leucine in 6 different ways. Now, if you replace position 54, the C by T, leucine is still leucine, so this for sure is not pathogenic. You don't have to investigate, you know for sure. Here's another one, it's a difficult one. Because here, amino acid serine at position 46 go to proline. It's completely different amino acid. So what happens? No one knows. Difficult to predict. And then we have a system to investigate E experimentally. So we take the gene and we change the code. So we replace the T by a C. And then we say, okay, try to make alpha-glucosidase. And when the cell does not succeed, it's a pathogenic mutation. And when we find a normal amount of alpha-glucosidase, apparently it's not a pathogenic mutation. Yesterday, we had presentation of the CRIM status in classic infantile pompe disease. It's important because CRIM negative tends to make high antibody titers and the therapy is less effective. So can we predict CRIM status? By and large, yes. But of course, you need to confirm in certain cases. I'll give an example. Well, here, for instance, it's not even pathogenic. So the CRIM status is yes, it's CRIM positive, and it's a simple prediction. And in this case, you have a shift in the very beginning of the protein. So in that case, the prediction is, no, you're not going to make any acid alpha-glucosidase. So this CRIM status will be CRIM negative. When you have such a defect or a stop, you know, it makes 40 amino acids. And then after that position, you don't make any. So you know for sure that this is CRIM negative, N. It's by prediction. But when you are at the very far end of the genetic code, you have made 99% of your acid alpha-glucosidase. So is this CRIM positive or is this CRIM negative? And then you don't know, and you have to investigate. And here, for instance, this deletion was investigated experimentally, and it is a CRIM positive. All right, and the other question you can ask, so now what about the correlation, genotype-phenotype? The truth is always better than the hypothesis. So let's look at the outcome. And then here we have made a prediction. For instance, when this mutation is very severe, we predict the outcome to be a classic infantile mutation. If the change is potentially less severe, it could be classic infantile, it could be maybe childhood. I've here listed it as childhood. Here, this for sure is less severe, experimentally investigated, we call it childhood, and this one is adult, but now I go to adult, and of course the best way to do is go to all the literature where all these patients were described and see whether these were patients with classic infantile or adult or childhood onset phenotype. That's a hell of a job, but in the Genzyme registry, the doctors, they put all this information in this registry. So that's an incredible important source, and now a group of patients, a group of people, they are investigating with the help of this registry what the correlations between phenotype and genotype are. But here, for instance, let my time. How am I doing, Nina?
None, zero.
I'm zero. Okay, so what I'll just have this. You have one, you have two copies. So you have one mutation in one, you have one in two, you add it up. Is one is not very severe and the other one is very severe, it's adult onset. That if the two are very severe, it's classic infantile. So that's the way we try to work it out. And there's one mutation, and I want to show this one because that's the next speaker. More than 90% of patients in our clinic have one particular mutation, and their ages of onset, they vary from below 1 till up to 70. And I want to end with this.
Thank you. So, the next talk is by Dr. Danesino about modifying factors. So he will explain why people with the same mutation have somewhat different phenotype.
Thank you, Arnold, for providing my slides. And first of all, let me thank Tiffany and the organizer for giving me the opportunity to be here and to share some of the results from our lab. The first slide shows some nice pictures of my university and of Pavia, and I hope that all of you will have a chance to visit our town. And I'm listing the people who are working with me on this data. The problem of clinical variability was extensively discussed, and you may recognize the name of the authors of this paper. And the statement which is relevant is that this variability can be brought about by secondary genetic or non-genetic factors. And so the enzymes or the mutations, as Arnold said, is not all. I think is a common experience for all the people working in the field to see a huge variation also within families. You see here a small group of families from our data, from Italian patients, and you see the huge variation in age of onset or in Walton score. Walton score is a score saying how much severely affected is the patient. So to explain this clinical variability, we can think, of course, about environment, but it is unlikely, and genetics. Other genes, variation in genes different from alpha-glucosidase, epigenetic modification which happens after genes have coded for proteins, or even, maybe it is a field which will develop in the future, somatic tissue-specific modification, so changes in genes happening not being not inherited, but happening after the birth of the child, anytime during the life, maybe in muscle cells. To address this topic, we went through the literature and we found a couple of papers by different groups about another storage disease like McArdle disease, and this paper states that there is a gene whose name is angiotensin-converting enzymes, and that some of different alleles of this gene may be related with different severity of the disease in that disease. What is angiotensin-converting enzyme? It's an enzyme which has apparently nothing to do with muscle function. It is working in the renin-angiotensin system, And as you may see here, it is related to problem of vasoconstriction or vasodilation. And when I'm saying that there is a polymorphism, I'm saying that in the general population, as all of us here, we may commonly found people who have variation in this gene. Some of them may carry an insertion, some more bases, 287 bases, and it is called E, this variation, or some of us may not have these 287 bases, and we call it a deletion, and the signal, the letter is D. It accounts for about 40% of the total variation of the level of activity of the enzyme. And here we can find something which becomes more interesting for our work. The insertion allele is associated with endurance, improved endurance performances in top sportsmen, and so something which has to do with muscle function. And the D allele is associated with higher percentage of type 2 fiber in the muscle, so structure of the muscle. This is just an example. There are many papers dealing with ACE and sports results, but in this example, you get this information. If you have a double dose of the D variants, if the D allele, then you will be able to climb only up to 8,000 meters on the Everest. But if you have a double dose of the insertion allele, you will be able to climb up to 8,500 meters. So it makes a difference. But going back to Pompe disease, when we measured and we checked these alleles in our group of patients, we observed that a double dose of the D allele was associated to quicker and higher changes in Walton score when compared to the other two alleles. Again, the presence of a double dose of the D allele, sorry for the arrows which moved, are associated to a higher chance of experiencing muscle pain. And the result that we like better is this one. We took into account the disease-free fraction, so the time of life till the disease becomes evident in the patient. And we saw that a double dose of the D allele was consistently associated with a shorter disease-free of life. Again, going through the literature, we find another gene. Again, there is a common polymorphism. Again, the gene is completely different from the previous one because now we are speaking about a gene coding, building a structurally protein for the muscle. And we have in the Caucasian population about 18% of the population carrying such a variation of this gene that the protein is not produced at all. Again, we see that the two alleles, R and X, are associated to different sport performances. And again, there is a report saying that the RR genotype is associated with the higher presence of type 2 fibers. This gene was also reported to be associated with muscle damage. Here on the right, you see that its contribution, I can move the arrow, okay, its contribution is not that much, but still you see that you may observe different levels of CK as the result of muscle damage in relation to the different genotypes. And again, some variation was observed in McArdle disease for the X genotype. So we did the same thing, and again, we saw that the XX genotype was associated to a much lower disease-free life. Of course, the next step was to try to understand if the two variations in the two genes can be studied together, and we did it. And we observed that a patient carrying a double dose of the D allele, the one which by itself alone was associated with the shorter disease-free life, when present in the same patient in association with the XX genotype, the second gene, and again alone was giving a shorter disease-free life, the two of them combined again gave a much shorter disease-free life. Our statistician, please do not ask me how he did, analyzed the combined data in a model, some kind of models, and he told us that there is no interaction between the two of them, but there is addition of the effects. And this fits with the starting point that the two genes have completely different functions. Unfortunately, the number of patients is quite small, but we checked the different genotypes for the two genes after 1 year of therapy by evaluating muscle mass variation. And you see again that we observed some variation in the different genotypes. Okay, with the DD genotype showing the worst results for ACE, and the RR genotype the worst, and XX and II the best result in both genes. And when we obtained this data, it had been shortly before suggested, I think it was a paper by Dr. Raben, that the enzyme replacement therapy was better in type 1 fiber, at least in the mice, and the two genotypes in which we observed the better result in muscle mass improvement are the two genotypes associated to an expected increase in type 1 fiber in men also. Then we thought that it could be interesting to look for other genes, and when our problem was to select the next one, and the next one was GYS1. The rationale for using or testing this gene is that genetic suppression of glycogen synthesis proved to decrease the storage of glycogen in the mouse model. But the results were not so good. Please note that here on the top right, you will see only 2 columns and not 3. The A2A2 genotype was not found because the second allele is quite rare, and so in the small sample of patients that we have, we did not find this kind of genotype. There is some difference, but not so much when CK is considered. We tried to combine GYS and ACTN3 as far as CK levels are combined. We observed some differences, but the results were not so good. So the point was to think in a different way to look for other genes which may be carried or harboring variations in relation to the variability. Again, you see in the first lines a paper suggesting that alpha-glucosidase can be a target for this Notch1 pathway. And so the idea was that, of course, alpha-glucosidase is relevant for his catalytic activity, and the loss of activity is the main cause of Pompe disease. But alpha-glucosidase is not alone, let's say it this way, and so it may interact with a number of different proteins. In this cartoon, you see a few number of interactions, but using this database, you may increase the number of interactions, but if you fill the slides, you will not understand anything. And so we went to collect some data in some cases. Here you see a long list of variations in the GAA gene in one patient, and please look only at the 3 polymorphism in exons. I can't work with the arrow, okay, where 3 polymorphism is written, because this DNA variation in the alpha-glucosidase gene will change the amino acid. These variations are additional to 2 disease-causing mutations that you see listed here. The IVS refers the -13, the common mutation in older patients that Arnold was talking before. And you see that in pink, there are the patients carrying a double dose of this variation. We are now trying to understand if this variation, I repeat, additional to the disease-causing mutation, may have any significance, and this is a tridimensional structure of the protein, and if we look at the same problem in a different way, you see the red spot on the left mainly, okay, the two red spots on the left, you see that these variations are on the surface of the protein. Why I'm stressing this point? They are far away from the catalytic site, so they are not relevant for the activity of the protein, but they are on the surface of the protein. And if alpha-glucosidase interacts with other proteins, this interaction is likely to be done on its surface. So we are now trying to understand if these amino acid residues are a point of interaction with any other protein interacting with alpha-glucosidase. We tried to, how much?
A couple of minutes.
Oh, okay. So we tried to use the same data on the polymorphism in relation with some other clinical parameters. And very quickly in these slides, we found something, but again, the results were not so good because only small differences were seen, maybe also because the number of patients is smaller. You see 6 minutes walking test and ACT entry differences between RR and the other genotypes. And another gene was investigated, which is again relevant in muscular performances and the type of fiber type, and here we had some better results in 6 minute walking test. In the last 2 or 3 slides, I'm very happy about these slides because it is a new work ongoing in my lab, but unfortunately, these data are not about Pompe disease, they are about a different disorder. But they can, I think it is a model, and it can be quickly applied to Pompe disease. What you see here is a very small selection of a database Excel sheet in which you see the result of exome variation in 8 patients with a different disorder, which is Schwachman syndrome. The colors refer to the different patients, so what you see in purple are 2 variations in the same patient. The database is made up of 10,000 lines. As you may imagine, it's useless to have so many information unless you are able to study them in a different way. In this disease, Schwachman syndrome, there is a severe pancreatic insufficiency and there are alterations in cell cycle pathway. So we were able to go down from, let's say, look at top right and left, patient number UPN 20. The number of variants we identified in him was 9,800, but the number of variants in the cell cycle pathway was only 36, and the variants in the pancreatic secretion pathways was only 42. So these are figures that we can deal with. And these are the results we like better by comparing in the 8 patients which variants were present or absent among them. We were able to build up this, which is called a dendrogram. This is about cell cycle. And you see that patient number 20 is far away from all other cases. If we take into account the other pathway, pancreatic secretion, you see it's completely different from the previous one, but patient number 20 is again different from all other patients. So the conclusion is that by studying these kind of variation, by studying the entire genome variation of the patient, also in a small group of cases, we are able to identify some patients which are different from all other patients, not in general, but refer to specific problems, cell cycle, pancreatic secretion. It could be done in Pompe disease, of course, for autophagy or any other set of genes related to muscle function. We are now looking for funding to repeat this kind of work in Pompe patient, but I think it's very promising because getting information of the general variation in all DNA of a patient by this way of looking at it, not in general, but targeting it with some specific muscle function, according to me, is promising. It may be helpful not only to understand clinical variation, but also differences in response to therapy and so on. Thank you.
Thank you very much. So the next talk is by Dr. Parenti on a subject which I think is very important, on biomarkers and Pompe disease.
First of all, I want to thank the organizers, the AMDA, IPA, and the House family for giving me the opportunity to be here, to join this meeting. and to talk about the work that we are doing in Naples, in Italy, in these two institutions, the Federico II University and the Teleton Institute of Genetics and Medicine. I will start with a short introduction. Some of the things that I will say have already been mentioned by other speakers yesterday or earlier this morning. So I will go through these introductory slides very quickly. And then I will come to the work that we are doing. Why we need markers? Markers are important for diagnosis, some of them. They are very important also to monitor, to understand the natural history of Pompe disease, and which is quite challenging because Pompe disease, like many other lysosomal storage diseases, is characterized by a broad phenotypic variability. Knowing the natural history, having good markers, reliable markers, measurable markers of disease, it's very important, it's critical to evaluate the efficacy of therapies and what are the benefits for patients, and to make therapeutic decisions such as when to start a treatment or how to adapt to personalize a treatment protocol. There are many different categories of measures, indicators. Clinical measures are very important, but there are also muscle pathology, imaging, molecular, immunological, and biochemical markers. And for each of these categories, there are several approaches that can be used. The hope is that combining all these approaches, we can get a complete picture of the status of the patients and what is the clinical course of the patient. Clinical measures have already been mentioned in some way. They are the cornerstone of our evaluations, and you have seen already this slide. There are over the years there have been a lot of effort to identify what are the best tests, best indicators like clinical domains, patient-reported outcome, other information. And it is also very important, it is to critically evaluate the relevance, the clinical relevance of these indicators. Yesterday we have heard about the glucose tetrasaccharide that is a biochemical marker of Pompe disease that has been shown to correlate with phenotype, with age in different groups of patients, infantile onset and late onset Pompe disease patients, with therapy and with the response of patients to therapy, with the highest values in patients that show minimal or no response to motor response to therapy. But there are possibly other markers that will come out. This is a paper that shows that myostatin, follistatin, or IGF-1 can vary over time, can be markers of disease. Molecular markers, Arnold already talked about the importance of the different mutations and on the phenotype of patients, and we have learned from the previous speaker that there are other factors other than the mutations of the GA gene that can impact on the clinical course of patients. And perhaps additional markers will come out from the analysis of, for instance, the gene expression profile in patients and animal models of Pompe disease. We focused on microRNAs. These are small RNAs. I have to thank Arnold for introducing those of you who are not familiar with these things, with the concept of DNA, RNA, and proteins. These are non-coding RNA. They do not code for proteins, so what is their function? They are transcribed in the nucleus, then they are processed, they are exported to the cytoplasm, they are further processed, and then their role is to interact with messenger RNA, the RNA that codes for proteins, And this results in a translational inhibition, a reduction of the amount of protein that is produced. So inhibition of the translation of messenger RNA and degradation of messenger RNA. They do not reduce completely the expression of a certain messenger RNA. It's not an on-off system. It's what? It is rather a fine-tuning of the expression of genes. And this is also because many microRNAs can interact with a certain genes, and also because this one microRNA can interact with different target genes. And the result of this interaction is a complex and intricate network of interaction between microRNAs and genes. Here you see that the same microRNA indicated with the same color interacts at different levels with different genes. This is the example of myogenesis, the production, the proliferation, and the differentiation of cells. And on the other hand, the same genes indicated by the circles can be the target of different microRNAs. And microRNAs have been proposed as markers of diseases in myopathies like myotonic dystrophy or Duchenne muscular dystrophy, and in many other pathological conditions like cancer. So what we did was to analyze in the mouse model that was generated by Nina Raben several years ago, the microRNA profile in tissues and plasma. These are some technical information about how we did it. And we analyzed the results by next generation, microRNAs by next generation sequencing, which is a powerful tool that allows sequencing of a huge number of sequences at the same time with the same analysis, and is quantitative. And then the results were analyzed by the bioinformatic core at TIGEN. And we did this in knockout mice compared to wild-type mice at two different ages that are representative of different stages of disease. And we are also doing the analysis at the later stages, the more advanced stages of disease in the mouse. And we collected a muscle, gastrocnemius, heart, and plasma. We didn't do the analysis on on other tissues because of the cost of this analysis. And these are the results. We found one microRNA that is differentially expressed at 9 months in the animals, 53 different microRNAs that are differentially expressed at 3 months in the gastrocnemius, 40 and 9 months, 42 additional microRNA that are differentially expressed both at 3 and 9 months, and in ART 9 that are differentially expressed at 3 months and 26 and 9 months. And I will not show all the results because it's 5-6 pages that are completely filled of numbers and analysis, mathematical analysis. This is what I show here is a simplification. Some of these microRNA, I expect you will not read all the numbers, but some of them, the message is that some of them, those that are shown in green, are down-regulated, and some of them are up-regulated and are shown in red. And the different shade of the colors indicates the level of differential expression. Some from a bioinformatic analysis and from what is known in the already known in the literature, we could identify that several of these microRNAs are involved in pathways like myogenesis, autophagy, atrophy, fibrosis, inflammation, cardiac hypertrophy. So this is in some way this supports the accuracy of our analysis. It was kind of expected. And here you see just a few examples. Some of these microRNA are implicated in controlling genes that are involved in more than a single pathway, and this is why the circles, there is some overlap between the circles. Just a few examples, myogenesis, here you see in red the microRNAs that are overexpressed, and in green those that are downregulated. Autophagy, the same, green downregulated, red overexpressed, and cardiac hypertrophy, again in normal or in pathological conditions. Then we looked at the target genes. We selected just a few, the 4 genes that are shown in red in the slide. These are validated from the literature. We know that they are target genes for the microRNAs that we have studied. In all cases, the microRNAs that control the expression of these genes are downregulated. So one would expect an increase in the expression of this gene. And although I have to say this cannot be completely true, because there are many RNAs that interact with the same genes. But in all cases, we found an increase in the expression of the target genes, which is consistent with the analysis that we have done. Now, the question is, can we use all this information to monitor patients? You may remember that one of the microRNAs was found differentially expressed in plasma. So a sample that is easy to collect from patients, it's MIR 486. So we asked for collaboration. We contacted other centers in Italy, Messina, Rome, Turin, Udine, and in the Netherlands, the Erasmus Medical Center. We asked Pim and Hans to give us some samples. And here you see a list of the samples that we have collected so far. NA stands for Naples, ME for Messina, Rome. You see that we have some information, some clinical information, but the information is not complete. You see also the definition of patients, the clinical, the phenotype is not really homogeneous between the different centers. We don't know anything about some of the samples that we have received. For instance, the samples that come from Turin, and we recently had also samples from Udine. These were the late onset patients, and these are the infantile patients that we got from Naples, from our centers, and from Rome. So in total, we have 43 samples. So far, we have analyzed only 31 of the samples. 8 samples are from infantile patients. We have analyzed 4, and 35 samples from late onset patients. And these are the results in plasma. This is what we found in infantile patients. There is a different level of these microRNAs in these patients. It is statistically significant. Of course, we have to analyze also other patients. And for late onset patients, again, we have statistical significance, but you see that there is a considerable overlap between controls and patients. And what we have to do next is to get more clinical information on these patients and try to find correlations between the levels of this microRNA and the clinical status of patients. Another question was, what happens if we give therapy, an enzyme replacement infusion? And this is what we have seen. For some patients, for 10 patients, we had samples before and after a single infusion. And you see that in almost all patients, there is a trend toward decrease, with the exception of 2 patients from Rotterdam. In one, there is stability, and the other one, there is a trend toward increase. So the first conclusion that we can draw from this preliminary, I would say preliminary analysis, is that we have found a distinctive profile in the Pompe disease mouse, that is distinctive microRNA profile, that we have found a microRNA that is differentially expressed in plasma, and this microRNA is also differentially expressed in samples from patients. Another conclusion is that we have to do a lot of work. First of all, to collect more information about samples and the patients that were involved in the study. We have information about age, gender, genotype, but we know almost nothing about phenotype, disease stage, effect of the response to ERT, for how long they've been treated with ERT, and so on. We are trying to just to collect the few data that we have, that try to find correlation. These are not results, I would say. This is just an attempt to get some correlation between what we know of patients and what we have found in our analysis. 2 of the patients were reported as affected by hyper-CK. They have low levels. We find high levels in the infantile patients, and the late onset patients are in the middle. So apparently there is some correlation from this first analysis with phenotype. We have information about age, and in the samples that we have analyzed, apparently there is again some information, some correlation between age and in the level of the MIR486 that we found in samples. Of course, these are late onset patients. In infantile patients, we find very high, much higher level. We also want to analyze other microRNA. We have found several microRNAs that are differentially expressed in tissues. We want to look, and some of them are relevant, are involved in pathways that are relevant for Pompe disease. So we want to select 4 or 5 other microRNAs and look directly in patients, not in the mouse. Then we want to analyze the effect of ERT in the mouse model. Here we can also look at what happens in tissues that we cannot do in patients. And perhaps we can, in the future, try to generate some systems that we can in some way manipulate, either by stressing the system or overexpressing one microRNA. Just to give you an example, we did something in fibroblasts. I know that fibroblasts are not the best cell line to study, but they can easily be manipulated. These are control and Pompe disease cell lines under basal condition. And if we starve the cells and we look at mirror